Bismillahirrahmanirrahim. Assalamu alaikum friends. Today we will discuss about soma clonal variation. So the name indicate of this specific topic that soma means somatic cell and clonal mean cloning and variation mean change occur during the tissue culture process. Now I want to define the soma clonal variation. So the changing occur in a plant or animal when we do tissue culture. So during the tissue culture time, some changes occur in plant. So this changes will be known as soma clonal variation. Now that is basically genetic variation when in the DNA of that specific plant the changes are occur, so we call them soma clonal variation. Now there are some types of soma clonal variation. The first one is a gametoclonal variation. So the variation observed among the plant regenerated from a gametic culture. When we take cells from gametes and do its culture, so the variation which takes place in those plants, we will be called them gametoclonal variation. Now the second type is androclonal variation. So the variation which observed due to enter or pollen culture. So when we take cells from pollen or enter and we grow them on tissue culture media, so the variation takes place is known as androclonal variation. Now gyanoclonal variation. So when we take cells from ovary and some variation when takes place in these, so they will be called gyanoclonal variation. Protoclonal variation, when we take cells from protoplast and grow them on a culture media, so that changes are occur, so they will be called protoclonal variation. And the last one is clonal variation, when we uh, do callus culture. So here some variation takes place, so that will be called clonal variation. So this is the short definition and some types of soma clonal variation. Now what is the causes of soma clonal variation? So the first one is mutation. So mutation is defined as any change occur in the nucleotide sequence. So in mutation, some variation are due to mutation. Some variation takes place in the plant during tissue culture. So they may either be insertion or deletion or a specific part of chromosome. So the first one reason is mutation. Now the second one reason is epigenetic. When methyl group are attached to a DNA sequence, so here the transcription will be not occur and gene will be not expressed. So is that why some variation occur in the tissue culture plant during the process of tissue culture. Now the other thing is plant regulate hormones. So we know that per plant growth, some basic hormones are involved. That is basically auxin and cytosine and gibberellin. So when the concentration of these hormone are changed and some abnormalities are occur, so some variation takes place because the number of chromosome will be increased, like the polyploidy, aneuploidy types are occur in the soma clonal plants during tissue culture. Now, how we can detect these soma clonal variation in plant when we do tissue culture? So, the first one is detection method is morphological detection. Suppose this is our main plant, and we take some cells from this plant and do its tissue culture. So here a plant will be produced. Now we do comparison between this plant and that plant. So we study the leaf size of the culture plant with comparison between the main plant, and we check the stem plant height. So this is the morphological detection for somatotonal variation. Now the second one is biochemical test. We do different type of test for this plant, like we check the concentration of carotenoids and concentration of chlorophyll or anthocyanin, which are basically the plant pigments, color pigments. So we check the concentration of these plant pigments, we do comparison with the main plant from which we do take cells. Now the third detection method is uh, molecular detection. So we know that first we extract DNA from this plant and also from this one, and these DNA we cut these DNA into a same restriction enzyme. After the cutting of restriction enzyme, we run these DNA on gel electrophoresis. So this is the main plant in which we can take cells from the part of the tissue culture. So this is the main plant fragments, and this is the culture plant fragments. So we cut these DNA molecule into restriction enzyme, and we run them on the electrophoresis gel. So here different types of band occurred. So if same band are occur in this electrophoresis, so the soma clonal variation will be not takes place in this plant. If the pigments are not if the fragments are not matching with each other, so the soma clonal variation will be takes place. Now the other detection technique which used for the soma clonal variation is PCR detection. So in this process we take DNA from our main plant, from which we take cells for tissue culture. So we take DNA from main plant and also take from the culture plant. So this is the DNA of culture plant, and we design a primer which are basically specific for this plant, for a specific gene. So the amplification will be occurred, and this DNA converted into 2 DNA molecule, and we use here the same primer for this specific gene which are we target in this DNA molecule. So here we also make a primer, which are basically the same primer. Now, when the amplification are occur and two DNA are palmed, so the soma clonal variation will be not takes place. And if the amplification or multiple copying of DNA are not occur, so uh, in this plant the soma clonal variation will be takes place. Now, in last we discuss the advantage and disadvantage of soma clonal variation. So the first advantage is they help in plant crop improvement. So when some changes or variation are takes place in the plant, so they help in the plant crops improvement. The second one is we can make different type of quality in plant using this somaclonal variation process. So we can make a salt tolerance plant, um, herbicide resistant plant, etc. So these are the advantages of soma clonal variation, that if we use this technique, so we can improve the crops production, and we can also improve salt tolerance plant, that they show resistance to salt high concentration. And we can also make herbicide resistant plant using this soma clonal variation technique. So the now and last we discussed the disadvantages of soma clonal variation. So the first disadvantage is making undesired quality in plant. So undesired quality will be occur in some time in plant due to soma clonal variation. So this is a lecture about today soma clonal variation, and inshallah see you in the next lecture. uh, I hope that you will be understand this topic. Thank you, see you next time.